Hello students. So welcome back to the channel. In this video we are going to study about the internal structure of Dicot stem. Now see one thing, this is your epidermis. Okay. Let us assume we have taken one TS and we are studying it. So this is epidermis which is bearing the multicellular hair known as trichomes. So I'm just drawing it 2, 3. I hope you can understand it. So this labeling I'm doing it, this is trichome. And this is your epidermis. Inner to this layer, then this next layer is your hypodermis. And in case of the dicot stem, the hypodermis is collenchymatous. Okay, in case of dicot stem, the hypodermis, which I'm drawing right now, the second layer is hypodermis, and this hypodermis is made up of collenchyma. Clear? Then the next layer, inner to the hypodermis, and between this next, I'm drawing it. This is your endodermis. I'm drawing it. Right now, this is endodermis. So this layer, which is left in between, this one, try to understand. This is your cortex. And this green color is your endodermis. So till now, we have drawn 4 layers. First is your epidermis. Second is your hypodermis. Third is your cortex. And fourth is your endodermis. Clear? Now check the diagram very carefully. See here. Now we have to draw vascular bundle. So I am drawing vascular bundle, and this line in between, it means it is cambium is present. 2 line I am drawing, it means cambium is present, and vascular bundle are conjoint, collateral and open, and they are present in the ring. Okay, vascular bundles are conjoint, collateral and open, and they are present in the form of a ring. So you can see this diagram, it is coming in the form of a ring only. Now on top of the vascular bundle, you will find this one, this layer. Check carefully, this is what I am doing. This is my pericycle, and the pericycle is the one which is made up of sclerenchyma. Analyze the diagram carefully. Please see it carefully, okay? So this is pericycle, and it is present in the form only on top of the vascular bundle. So it is known as bundle cap also. And it is present like a semilunar patches. So this layer, this white I am drawing it separately, you can understand. This is pericycle, and pericycle is sclerenchymatous. So you know very well that simple permanent tissue are of 3 types: parenchyma, collenchyma and sclerenchyma. Cortex is parenchymatous in nature. Hypodermis is collenchymatous in nature. Pericycle is sclerenchymatous in nature. Is it clear? And complex permanent tissues are your xylem and phloem, which is your vascular bundle. The vascular bundle means which include both xylem and phloem. Okay. And vascular bundles are of which nature? We have already studied. They are conjoint, they are collateral, and they are open. Open means cambium is present. Up till here, I hope topic is clear. Now, one important point. See, in case of the dicot stem, endodermis will store starch. So we say it is having starch sheet. It is a starch sheet. Pericycle is present as a sclerenchymatous only on top of vascular bundle. So we say semilunar patches. The semilunar patches are made by the pericycle. This much is clear. Now, see, The center path, this area, which is center area, okay, the center area is known as pith. This is known as pith, which is made up of again parenchymatous tissue. This is pith or known as medulla. And if you see, this is making a rays. Check my diagram, I'm making a rays, so we will say these are your medullary rays. So center, if they are present in the center, this area right now I'm doing it, this is pith or medulla. And when they are extending between the vascular bundle, that time we are calling it as medullary rays. So this is again parenchymatous in origin. So I'm just doing the labeling, try to check my diagram. This is known as pith or medulla. And this part, okay, this part I'm writing it here. This is known as medullary rays. So this is the short diagram. Now we have to study properly in detail. Just give me just 1 minute. Now we will do one thing. We will cut this part. We will cut this part. So I hope you are able to understand. I'm taking only 2 vascular bundles in my diagram. And now we will be studying in detail large diagram of this Dicot stem. Okay. Now try to match it. This is your epidermis, which is single layered, which can be covered with the cuticle, because cuticle can be present in the stem anatomy as well as in the leaf anatomy, but cuticle is always absent in the root anatomy. And it is having trichomes, which are multicellular with multicellular, and they can be branched also. So I'm just drawing 2-3 trichomes. And NCRT has told that they can have few stomatas also. So stem in the NCRT, if you check carefully, they are telling it is actually having few stomatas. So this is cuticle. This is your trichome labeling. Okay. And this is your epidermis. So I think so first layer is clear. You know very well, work of epidermis is for protection. The epidermis is meant for protection. And trichome will also help in preventing the loss of water due to transpiration. Now, 3 layers, that is hypodermis, cortex and endodermis, they are the part of the cortex. The cortex is divided into 3 regions. So first of all, I'm drawing hypodermis right now. In your exam, you have to draw it this way only. This is the 2-3 layers you have to draw. Right now, 1 layer I've drawn. This is second layer in progress. To draw at least 3 layers, and after that you have to do the deposition of the hemicellulose and pectin at corner. So deposition has to be done of hemicellulose and pectin at the corner. Is it clear? Because you know very well that this is your living mechanical tissue. So intercellular space should not be visible, and you should do the deposition of which chemical? Hemicellulose and pectin, because this is your living mechanical tissue and it is going to provide support in the young stem as well as leaf, petiole, your flower, stalk of the flower. So they are providing the support there. So this is the way the angles should be filled, because this is your angular collenchyma. After this, you have to draw. Next layer is your cortex. Means you have to draw parenchymatous cells, so you can leave intracellular spaces here. You can leave intracellular spaces here because they are parenchymatous. So 2-3 layers it should be. Then next you have to draw endodermis. So for endodermis, I have used green color, so I'll be using the same color here. This is endodermis. And endodermis is storing starch. So it is also known as starch sheet. So endodermis is storing starch, so we call it as a starch sheet. Is it clear? Now next layer will be of the pericycle, which is present as a form of semilunar patches over the vascular bundle. So first of all, let us draw two vascular bundle because we have taken from the two only. So these are the vascular bundle drawn. Then we have to draw cambium because vascular bundles are conjoint, collateral and open. This part has to be filled with the phloem area. So this is the phloem area. Phloem is present only on one side. And then xylem, we have to focus ki xylem is actually endark xylem with centrifugal growth. And now in between we have to draw medullary ray cells. But here on top we have to draw this area, check carefully, this area, this much we have to draw as a pericycle, which is in the form of a sclerenchyma, which is making a semilunar patches or bundle cap. Double wall indicate the cell is dead. Okay, double wall indicate the cell is dead. Because of the lignin deposition, you know very well, when the lignin deposition is going on in the secondary wall, then the center is nothing is left. We say lumen is there. And now we have to fill the remaining area with this medullary ray cells. These are the medullary ray cells, which are again parenchymatous. So the Dicot stem is the one in which you study all types of the permanent tissue. Simple, all three types of simple permanent tissue we study here, and we study the complex permanent tissues also. Complex permanent tissues means xylem and phloem. We have to make it more pointed. Slowly we have to go for pointing, okay? Because we have taken this part as on this 50 years. So this is the way how to draw the diagram. And when you draw the diagram, once you draw the diagram, then you can understand easily the secondary growth also. So let us match all the labelings. This is your epidermis. Then this is your collenchyma hypodermis. Then next, this is your cortex. Okay. This was your endodermis starch sheet. This is pericycle, which is made up of sclerenchyma. This is pith or medulla. And these are your medullary rays. So I hope this video in which I have drawn the diagram in front of you and I have given you the tips how to draw the diagram will help you to understand the concept very easily. Thanks for watching.